Bienvenidos, Wildcats. Welcome back for another exciting topic in biochemistry. What we're going to start on now is a couple of topics in a row that talk about hemoglobin. Hemoglobin is the protein in our blood that transports oxygen from our lungs to our tissues and also brings back, we're going to get rid of our carbon dioxide. So what we need to do is to first look at the structure of hemoglobin and myoglobin, and then once we understand the structure, we can start looking at the function. So we'll just start off first looking at the structure. And so what you see on this first slide, this is the heme group. I mentioned it in that original protein structure topic not too long ago. And what I'm showing you here is the difference between what we call fresh meat. This happens to be beef from a cow. And then this is aged. And so lots of times people say, oh, I got aged top sirloin steak. What does aged means? It means that you kind of left it out for a long time. So what are we looking at here? We're looking at the color, okay? And so what we see is we see a bright red color here, and then we see a dark color. So why is that? Well, that has to do with the iron going from Fe2 plus to Fe3 plus. So it's the oxidation of iron that actually changes the color. And so when you look at a piece of meat and you go, oh, that meat looks brown to me, I want you to be thinking about the iron, and I want you to be thinking about the hemoglobin and often the myoglobin as well. So what is the protein doing? What is the hemoglobin, which is a tetramer, is taking 4 molecules of O2. And myoglobin is in the muscle, and it just transports 1 molecule of O2 at a time. So let's go ahead and get started. So we have 3 key concepts in oxygen transport. The first one is myoglobin, which is in the muscle, and myoglobin is a single subunit. So it's a monomer, and it only carries 1 O2 molecule at a time. And then hemoglobin is a tetramer, and that tetramer can carry 4 O2 molecules, and it's made up of a 2 alpha and 2 beta, so it's an alpha 2 beta 2 tetramer, and we'll talk a lot about how that works. And then we'll talk about how oxygen binds to hemoglobin. That again is the major protein in our red blood cells. And then we're going to look a little bit deeper at how does that structure of that tetrameric hemoglobin protein change in the presence and absence of oxygen. And what you're going to see is that it's a very efficient O2 load, and the O2 loading means that it takes oxygen from the lungs and brings it to the tissue. So we'll talk about how that works. Okay, time for a movie. So as we say, it's movie time. Enjoy.
Hemoglobin, a major protein found in red blood cells, is responsible for transporting oxygen from the lungs to tissues throughout the body. Hemoglobin is a tetramer made up of 4 polypeptides, 2 alpha subunits and 2 beta subunits. This tetrameric structure is essentially a dimer of alpha-beta heterodimers. At the core of each subunit is a heme complex made up of a porphyrin ring with a central ferrous iron atom. The iron has 6 coordination bonds, 4 with nitrogens in the plane of the porphyrin ring, and 2 with histidine residues above and below the ring, called the proximal and distal histidines. In the absence of oxygen, the heme group is puckered because of the large radius of the ferrous iron atom. Oxygen binding to the heme group reduces the radius of the iron, forming a planar heme. Because of the bond between the iron and proximal histidine, movement of the iron moves the histidine, which in turn moves a helical segment called the F helix of the hemoglobin polypeptide. The F helices are located at the interface between subunits, and therefore the movement of the F helix of one subunit can impact the structure of the entire protein, shifting the hemoglobin molecule from one conformation, called the T state, to another conformation called the R state. Oxygen acts as a positive allosteric effector for hemoglobin by shifting hemoglobin from the T state to the R state. Several other molecules act as negative allosteric effectors. Their binding shifts the conformational equilibrium towards the T state. The small compound 2,3-bisphosphoglycerate, or 2,3-BPG, traps hemoglobin in the T state by binding between 2 beta subunits in the center of the hemoglobin tetramer. In this way, 2,3-BPG acts as a classic heterotropic negative allosteric regulator. Binding of 2,3-BPG to a secondary site prevents binding of oxygen to the primary site. 2,3-BPG plays a role in facilitating oxygen transport from maternal cells to fetal cells during pregnancy. Although 2,3-BPG is present in both adult and embryonic red blood cells, fetal hemoglobin consists of a tetramer complex in which the normal adult beta subunit is replaced by a special fetal hemoglobin subunit called gamma. The fetal form of hemoglobin has a lower affinity for 2,3-BPG. As a result, fetal blood cells have more hemoglobin in the high-affinity R state conformation compared to maternal red blood cells, and therefore fetal hemoglobin can acquire oxygen from maternal hemoglobin. The multiple modes of hemoglobin regulation ensure that oxygen is efficiently delivered to tissues. Hemoglobin also serves as a classic example of how protein structure mediates protein function.
All right, back from the movie. Again, hopefully that gave you more of a sort of visual effect of how this works. One of the things you might have noticed is I talked a lot about the R form and the T form. So R form is going to be the form that binds oxygen, and the T form is going to be what we call the deoxy form. And so we're going to spend a lot of time about how those two conformations go through their equilibrium as more R form, which is in the lungs, lots of oxygen, T form in the tissues where it dumps the oxygen, and myoglobin is often going to pick that up. So again, myoglobin is a monomer. And so we did a lot of studies, biochemists did a lot of studies on myoglobin first. In fact, that was that protein structure that I showed you because it was easier to understand. But really in terms of how we get oxygen, we need to look at the hemoglobin. It's called a heterotetramer that I just put on the last slide. It's 2 subunits of the alpha subunit and 2 of the beta. So it's an alpha 2, beta 2 heterotetramer. And each of those subunits has 1 heme group with an iron that binds O2. Okay, so they're structurally related. So if we go over here, we're looking at, again, the 3-dimensional structure, which was obtained by X-ray crystallography. This is the myoglobin that I showed you in the previous topic that John Kendrew was able to come up with in a crude way. Then if you look at the alpha subunit of the heterotetramer hemoglobin, you can see that the helices are basically in the same place. So we'll talk about this in a couple of topics, the evolution of the globin gene. So there was an ancestral globin gene that gave rise to myoglobin, alpha globin, beta globin, and also the gamma globin, which is the fetal form of this. And you can tell I've lined these up, used a computer and rendered them. So I have my heme group in basically the same place. And what you can tell is that all of these protein structures are similar. And so, as we said, in many cases, but certainly not every case, the structure of the protein can be conserved for the same function. So this would be a case where we have same structure, same function. And so this one worked out quite nicely because all of them are oxygen binding proteins. And again, don't be fooled. We did the one with the steering wheel and the skateboard wheel. It kind of looks the same, but it's different. But in this case, it is the same structure and the same function. So that's going to be good. All right. So how does this oxygen transport work? So let's look at it. So basically what we have here is we're going to look at this in more detail in a minute. But this is the helix, and this is the histidine residue on the E helix, and it's a number 7 position. So there's a histidine sitting in towards that heme. And so here's the histidine residue, which you should recognize. And then there's another histidine residue on one of those other alpha helices. This is the F helix, and it's position number 8. And so this is, has a histidine that's pointing in towards the heme. So here's the heme group. So this is called the proximal histidine, and it's making a very strong interaction with the iron. And then the iron is what is going to form a strong interaction with the O2 ligand. Now, what the distal histidine does is it stabilizes that oxygen so that it can be transported from the lungs to the tissue, and it forms a hydrogen bond between the histidine and that oxygen, and it kind of holds the oxygen in there. And then as the red blood cell travels through your blood, and this hemoglobin molecule finds itself in the tissues, like in a muscle tissue, the affinity of the oxygen for the iron is going to change because what you'll see is the hemoglobin undergoes a conformational switch, and what's happening is that these helices begin to move. Okay, so and we'll talk more about that. So this is what's going on. So what you want to think about is really it's one of these things where I use my hands again, and so we're going to have the R state and the T state. And so what causes that conformational change? So what you see here, this is my proximal histidine, which is the F8, and it forms a very strong bond with that iron. And so what you have in the deoxy, so this would be in the T conformation, there's no oxygen here. And the iron is the iron van der Waals is a little bit larger, and the heme is puckered. So the iron is not in the heme because it's too large to fit in the heme. And the proximal histidine is in a position here. And then what happens is when oxygen binds, is it pulls the iron towards the oxygen. So it's pulling it. And when that happens, what happens is the heme is going to pull because the iron is now in the plane of the heme. So when the iron moves, the histidine moves. And when the histidine moves, the helix moves. So the helix actually moves. And when that helix moves, it changes the conformation of the whole protein. So again, it's the deoxy state. The iron is too large to fit in the plane of the heme. And then when the oxygen binds, because of the electron sharing and the electronegative interaction there, what happens is the iron radius gets smaller. It can fit inside the heme, and as it fits inside the heme, it pulls the F helix, and as it pulls that helix, it's going to change the conformation of the protein. Okay, so then in the last slide here, let's just kind of pull it all together. And what we have, it's easier for me to show you over here. These are the structures, again, from X-ray crystallography in the deoxy state. So notice here's this is my proximal heme and my proximal histidine, my distal histidine. This is my heme, and this is my iron. And so there's no oxygen here. So it's deoxy, and the F helix is in this position. Then when the oxygen binds, the O2 binds, it's stabilized by the histidine in the distal helix, or the distal histidine. And again, it pulls that iron into the plane of the heme. And as it does that, it pulls the helix, the F helix, towards the heme. And when that happens, this whole helix begins to move. We can then overlay both the deoxy and the oxy so that we can get a better idea of really how much movement is there. And this is really what you see right here. That's the distance of about 0.6 angstroms of a movement between the deoxy and the oxy, and that's pulling. So now I have that movement. So this was all captured by X-ray crystallography using the structure of the deoxy and the oxyhemoglobin. So that does it for a quick overview of the structure and binding of oxygen to hemoglobin in this tetrameric protein. Hasta luego, Wildcats.